Hello, welcome to the presentation. First of all, I'd like to introduce myself. My name is Professor Graham Williams. I'm a Professor of Forensic Biology in the School of Law, Policing and Forensic, and I'm a member of the Centre for Crime, Justice and Security. So in celebration of National DNA Day, I want to answer the commonly asked questions about forensic DNA profiling. So my background is a forensic biologist. I've been a practicing forensic biologist in 2002. I've done quite a few cases involving DNA. And since 2007, I've been research active in the field. And I would like to think I have quite a bit of knowledge about forensic DNA profiling, and I really hope to ask you any questions that you may have. So the first one is often asked is, what is DNA? Well, DNA stands for deoxyribonucleic acid, which is why everyone calls it DNA. So this is basically a complex molecule which holds the blueprint of life. The DNA is thought to be unique, so the whole strand of DNA is thought to be unique unless you're an identical twin. However, with forensic DNA profiling, we don't profile the whole DNA, so we're not necessarily unique. This DNA is present in nearly every cell in the body, and it's the same. So for every nucleated cell, we have the same DNA profile, whether it's a blood cell, skin cell, or a hair follicle cell, or a sheik cell. It holds the same DNA profile. However, one of the things about the DNA is in everyone who's human, most of our DNA is the same. So quite a large percentage of our DNA is the same between everybody. So in forensic DNA profiling, what we do, we target the part of the DNA that are different. So there's no point in going after the DNA that's the same in everybody, but it had no discriminating power. So what we go after the bits that are different. In other words, the hypervariable regions. So how is DNA profiling done? Well, the number of processes involved, a number of steps involved in this process. So we start off with maybe a body fluid. So let's say some blood has been left behind at the crime scene. So the first thing is that blood has to be sampled and recovered from the crime scene. So this can just be a swab. So the swab sample will be taken, placed inside a tube, and then the tube is sent off to the DNA laboratory, and the work product for a time provider or a DNA analytical laboratory. So when it gets there, the first thing it undergoes is, well, first an assessment of the swab. So what did it look like? Did it look like blood? You might test it for blood to confirm that it is blood. But in terms of DNA, what the first thing we do is extract the DNA. So what we do is take the swab head and cut the swab head off, and then we put it into a tube and contain it and fill it up with a buffer. So typically something like PBS, which is phosphate buffer saline, and we agitate it. So what this does, it gets all the cells and gets it loose into the liquid. And then what we do with lysis, so lysis or lysis is the act of breaking down the cells. So the DNA is inside the nucleus of a cell. So we've got a cell membrane inside the nucleus of the cell, and the cell all might be packed together. So it's all got to be broken apart to get to the DNA itself. So this is called the lysis step, and that's part of extraction. So it can be either heat treated, we could rupture them, but typically we might have a lysis buffer, and that will rupture the cell membranes. It can also be something like detergent, just rupture apart, and that what we end up with is some sort of a slurry, if you like, some sort of soup where DNA is floating around in it. Sometimes you can analyse straight from that, but for forensic purposes, we want to purify this. So what we then do, we run the DNA through a silica filter, so like a spin column, and silica with a silica membrane. So in a high saline solution with a silica membrane, double-stranded DNA will bind to it, but nothing else will. So you put it into a high saline solution and you run it through, and all the DNA will bind to the silica. And then once it's all run through, we'll wash it off. So anything that's attached to the DNA will get washed off. And then what we do, we remove the high saline solution. So you can just run it through water, for example, and all the DNA will drop off, and that will go into a liquid. So what we have is a purified sample. So following the extraction, we then have to quantify it. That means we measure how much DNA is present, because when we go on to the next step, we need to know how much If we've got too little, we won't get a good enough DNA profile. If we have too much, we get a very messy profile. So we want to get it just right. So do a quantification step. And all we do is just measure the DNA alongside a known standards. And so once we quantify that, we then amplify the DNA using PCR, or polymerase chain reaction. And this is a way of amplifying small amounts of samples up into large amounts quite rapidly and accurately. And so this gives us enough to actually characterize. And following amplification, we then do something called electrophoresis. So you've got all these fragments of DNA, and what we want to do is now separate them out according to size. And so we run through electrophoresis, and this is normally some form of polymer. So we do something called capillary electrophoresis, where you have a very, very thin capillary with a polymer through it. And the smaller the fragment of DNA, the faster it travels through. So the smaller fragment of DNA will travel through first, and at the end of the capillary we have a laser. And that laser will basically shine on the DNA and of course fluorescence, and we can detect that fluorescence. And so the quicker it goes through, the smaller the fragment, and then we end up with peaks of the DNA, and from there we can get a DNA profile. So what did the DNA profile actually look like? So quite often we'll see images of bands. In fact, I was watching Line of Duty last night, and they showed a DNA profile, and they had all these bands. We've not used bands since the 1990s in forensics, so it's all been peaks rather than bands. So that was very much out of date. I nearly threw my my shoe at the TV screen when I saw that. So a DNA profile actually looks like this. So this DNA profile is mine. So what you're seeing there is my DNA profile, and what you're seeing is these areas here like this. And we've got all these different areas, and here we've got amelogenin, and what you've got here is SY, so that indicates that my sex is male. If I was female, or like biologically female, I would just have X here, but it would be twice as high. I'd have a very long P, but I wouldn't have a Y. Because a female, someone who's biologically female is XX, someone who's biologically male is XY. But what you see is each of these locations, you've got two pairs of peaks, or sometimes you've got one peak that's extra large. So each of these peaks, one of these peaks is inherited from my mother, and the other one from my father. I don't know which one, but that's what it tends to do. But if I've got one peak, which is higher, so I've got one peak here, two peaks here, you can see this one peak here is higher. And that's because I've inherited the same DNA from my mother and from my father. So I've got a double weighting of that DNA. That's called a homozygote. If they're different, called a heterozygote. You can see underneath of each of these peaks, you've got numbers. They've got 13, 15, 15, 18, 11, 12. So these numbers here is the DNA profile. So it's not about the peak, it's about these numbers here. So my DNA profile, were I to read it out, would be 13, 15, 15, 18, 11, 12. And I'll compare that with somebody else's DNA, or a DNA from a crime scene. If all the numbers are the same, then the DNA profiles match. If one number is different, then the DNA profiles don't match. So it's an all or nothing rule in place. So looking a bit closer in, so I've zoomed in, so you can see the amelogenin got SY, and then got 13, 15. And these numbers here refer to the marker. So D means the DNA, 10 means the 10th pair of chromosomes, and sequence 1248 refers to a specific sequence on that chromosome. So it tells us where that location is. So what is a DNA sample? So a DNA sample is any fluid or tissue that could contain DNA. So it can be blood, saliva, semen, vaginal fluid, skin cells, or hair. And these can be in form of a swab, it can be a liquid in a tube, it can be a stain on the fabric, so for example on clothing, it could be a lump of tissue, for example. So a DNA sample can be anything that could, in theory, contain DNA. But sometimes it can be speculative. You might swab something hoping to find DNA, but there's nothing there. but just you won't find out until you test it for it. So how is DNA profiling used to identify someone? Well, if you get a DNA profile from a crime scene thing, so if I go to a crime scene, there's blood. I get a DNA profile from it, I've got all these peaks of them. And then I've got a DNA profile obtained from an individual. So let's say we've got someone being beaten up and there's been a fight between two people. Both people are bleeding and there's blood that's been left behind at the scene which didn't match out the victim. So we've got that DNA profile. The police may be speaking to some various witnesses and they might identify someone being Joe Bloggs. So they then go and arrest Joe Bloggs and get a DNA profile of Joe Bloggs. So what I've got two DNA profiles. Okay? If all the numbers match, so I've got 13, 15, 15, 18, 17, 20 and so on. If all the numbers are the same, then the DNA tested could have come from that individual. If the numbers are not the same, if they're different, then the DNA tested did not come from that individual, and that is a conclusive elimination. The DNA profiles are different, then it could not have come from that person. The DNA profile is the same, then it could have come from that person, along with anybody else who had the same DNA profile. So what about identical twins? Well, if you've got identical, or what we call monozygotic twins, they can have the same DNA profile. So whilst these are rare, there have been cases where the suspect was one of a pair of identical twins. So if the DNA evidence is the only one in the case and it implicates two people, but you can't tell which one it is, now you can't progress that. So you can't imprison both people, knowing it's one of them. So you can't differentiate between them, then it can't go any further. Okay? And there's been a number of cases. There's been a couple of cases in Britain, and there's one very high-profile case in France, and where that was the issue. And both cases were rape cases. But however, there is research out there that's shown a number of strategies for differentiating between identical twins. So these can be mutation analysis. So one twin may have a new mutation that the other one doesn't. There's also an epigenetic approach where one twin might be exposed to a different environment to the other, and that has made some slight changes to their genome which we were able to detect. So there are strategies to dealing with that, but from a DNA profiling point of view, then if they're truly identical twins, then their DNA profiles will be the same. So what information can you get from a DNA profile? So sometimes people worry about, what secrets are being held on the database. Can we get your medical information? So a DNA profile in itself, the power behind the DNA profile is comparative. It's comparing your profile with that of the crime scene saying, it will get the National DNA Database, is your profile on there? That's where the power of DNA profiling is. It's comparative. There's very little intelligent value that can be obtained from a DNA profile. Some information can be obtained, but nothing that can't be obtained from standard methods. So for one, we can identify the sex of an individual. We can say from a DNA profile whether that person is male or female. I'm talking about the sex, which is the biological aspect of it. Obviously, whether this correlates to gender, so gender, we know, can be very fluid. So some people can be born male and identify female later on. However, biologically, they retain the same sex. So which means that someone who was born a male and then made a choice or became female later on or identified as female later on and may have the outward appearance of female, they will still have a male DNA profile. So in terms of intelligence, if I get a DNA profile, am I looking for a male? And that may not always be the case. So that's the intelligence value there. So we always said this had the thought of the DNA of the male sex, and this may not necessarily mean a male gender. We can also do racial inference. So some of the DNA profiles and some of the numbers that we get, which are called alleles, are more common in some races than others. So as a result, providing we have the correct database to compare it with, we can make an inference as to whether someone could be Caucasian, Afro-Caribbean, or Asian, and we can make an inference. It's a probabilistic assessment. This will be intelligence only. And also through the use of population database, we can also have an inference as whether someone got red hair or not. So for those who have the gene for red hair, some of the genes that we detect are more common in those with red hair than not. So that can only be gleaned if we have an appropriate population database to compare it to. So from a DNA profile itself, I'm on the screen, so like the one I showed you earlier, all I can get from that is just text, and that's it. I can't get anything further, and that's from the profile. So people do express liberty concerns about the National DNA Database, saying we could get medical information, we could get this and that, and the fact is we can't. There are samples which, should they be tested for, may get that information, but in terms of DNA profiling and the information held in the National DNA Database, there's nothing more we can get from that other than what we can do from the use of databases. So how long did DNA stay on clothes? So this is quite a common question. So if I were to go and beat somebody up and I get the victim blood on my clothing, how long will that stay on my clothes for? The question is, the answer is how long is the piece of string? It depends what they do with it. Because once blood is dried, it's very difficult to get out. I could launder it, I could do this, I could do that. If I launder it before it gets dry, that has an effect. It's thinking about the ambient temperature, it is humid, it is hot, it is warm. So it's really, really difficult to say. So obviously the longer you leave it, the less likely you are to get DNA through removal or simple degradation. But I mean, I can't give an answer on that one because it's just so many factors affect the persistency of DNA on clothing. But if you said to me, okay, someone was beaten up, we've identified the suspect, we've identified the jumper that he was wearing at the time, and it was a week later, would you expect to find his blood on that jumper? And I would say yes. However, had it been laundered, what happened to that clothing in the meantime, and that kind of thing. And they say, oh, as far as you're aware, we recovered it from his laundry basket, it's not been washed yet. And so 7 days, not being washed, yeah, I would expect to find blood. So there's many factors into play coming into this aspect. So how long did DNA survive? And again, the answer is how long is a piece of string. So it's all very many factors into play. So DNA can be obtained 30 years after. It all depends on how the DNA is stored, how the DNA is kept in. Sometimes you might hear in the news of DNA being recovered after thousands of years. So in terms of archaeological samples, that DNA tends to be mitochondrial DNA. So whereas in forensic DNA profiling, we use nuclear DNA. So nuclear DNA is a lot less stable than mitochondrial DNA. So mitochondrial DNA is smaller and circular, so it didn't fall apart, such as nuclear DNA, which is long and linear, so it can fade, the engines are coming apart. So in terms of forensic DNA profiling, 30 to 40 years, I would reasonably expect to get a DNA profile if it's been stored in the right conditions. But I certainly wouldn't automatically say no if I said the item was 30 years old. I said, well, let's look into it, and it may be possible. Can you tell how long DNA has been there? So this is a question being asked, not only just by people in general, but also by police officers. So I've had a case where I've got a DNA profile of the gun, and it matched Joe Blogs, and I reported that to the police, and the police have said, oh, that's brilliant. Can you say how long it's been there? And I said, no, I can't. I can't add a time aspect to the DNA. So it could have been there yesterday, it could have been put there 10 years ago. There's no way for me to test for that. So that can be a bit of a problem sometimes. So, for example, if you had a nosebleed at a crime scene, but the crime's not happened yet, and then 2 weeks later a crime occurred, your blood is at crime scene, I can't tell whether it's from the crime or from whether it's earlier. So it's very difficult. However, having said that, there's a lot of research out there that's working on that, and there's some promising research coming out which may actually be able to do this. And so that would be really, really useful. So how did the National DNA Database actually work? So basically what happened is the two main types of staff profiles are held on the database. One is from individuals, so anyone who gets arrested in the UK has to give samples, has to give a DNA sample, as well as fingerprints and photos and so on, and by law. And so that sample then profiles, and that profile with a string of numbers is uploaded onto a database and is kept on there. The other type of profile that's put on the database is something called unsolved crime things. So if you go to a crime scene, get a blood sample, you get a DNA profile, you then search it against the National DNA Database, but it didn't match against anybody. So what you've got is a DNA profile of someone who is not yet on the National DNA Database. So you put that on the database, and that's an unsolved crime thing. So what then happens is, let's say 2 weeks down the line, a crime occurs, you get a DNA profile, you then upload against the DNA on the DNA database, and if you've been arrested and your DNA profile is on there, it will match against you. So the police will then get a match report saying a DNA profile that came from his clothing and it's matched against Joe Bloggs. The police can then use that to go and arrest Joe Bloggs and ask him, why did we find your DNA there? Okay. So they can't charge on that alone. So National DNA Database matches is intelligence only. It's not evidential. So someone can't be convicted on a database match, but they can be arrested and investigated as a result. And in terms of the unsolved crime thing, so if you've got a crime scene thing uploaded it, didn't match against anyone, you can just leave it there. And then let's say 3 years down the line, you get arrested for something else, something completely unrelated. You get your DNA profile taken, you get a sample, profile, put on the database, it will match against an unsolved crime thing, and then you'll be flagged up for that. So that's how it works. So it's all about comparing. It's just a massive database with a string of numbers, and it just matches, and it tells you whether it matches you or not. And it's intelligent. So what you do is give the police, you have a suspect, go and look at this person. So what are human rights concerns about the DNA database? One of them is about the information that's available. So what information can we get from a DNA profile? And I said before, there's very little. So there's some privacy concerns about that. But one of the main concerns about the DNA database is that you get a DNA sample taken if you're arrested, but it says nothing about being charged. So you could be arrested, and then as part of the investigation, it turns out that you've got nothing to do with it. So you arrested a suspect, they've questioned you, you've answered their questions sufficiently, and they're now satisfied that you have got nothing to do with the offence whatsoever. Your DNA profile is still on the database. So that's some of the concerns. Also, if you've been arrested, you've been charged, you go to court, you have a trial, and you're found not guilty, your DNA profile is still on the database. So that was a concern. So a few years ago, changes were made that if you want to have your DNA profile removed, you can do. You just need to write a letter to the arresting police force and ask for them to remove it for you, and then that can be done. Only if you either haven't been charged or you've been found not guilty. But each force has its own procedure for doing that. So if this is something you want to do, contact the police force that arrested you and look on their website, and there's normally a way of doing that. Can my DNA be used to identify my relative? So this is a common question. So it's been really high profile. So if my DNA profile is on the database, can it be used to identify my relative? And if we mean blood relative, then yes, it can. Because if all the numbers match apart from one, they've got a string of numbers, they've all got to match, they all got to be the same to match your profile. But if one number is different, then it's not yours. But if there's only one number different, then it's probably from someone who's closely related to you. So it could be your brother or sister. So what we can do, if we have a DNA profile, we upload it to the database, it doesn't match, we can do something called a familial search. And so what we do, we put the database or your profile up onto the database, remove one of the numbers and replace it with a wildcard, and then see if it matches. And then we keep doing that with each number until we get a list. And then what you end up with is a list of names who people may be closely related. So if you get somebody who's got maybe 95% of your profile the same, which is 5% different, what we might do is go to that person. So let's say that person with James Blogs. So we go to James Blogs, and the police will go to James Blogs and say, right, we're not interested in you because the profiles do not match. However, the profiles are very similar, so we'd like to know who your brothers and sisters are. And if the DNA profile indicates it's a male, we'd say, we'd like to know who your brothers are, and then get profiles off them and see if any of them match. And so that's the way DNA can be used to identify your relatives. So, do I have to give a DNA sample when arrested? Simply put, yes. When you're arrested, you are required to give fingerprints and you're required to give a DNA sample. If you refuse to give a DNA sample, they can take it by force. So you are required under the Police and Criminal Evidence Act to provide a DNA sample when arrested. If I'm arrested for rape, do I have to give a semen sample? So this has been a fairly common question. So in a rape case, the DNA comes from the semen. So the semen sample has been recovered and profiled, and the DNA profile obtained. So if the profile came from semen, do I then have to give a semen sample? And the answer is no. Because your DNA profile will be the same regardless of what body fluid you give. So this is something called body fluid concordancy. So the DNA profile will be the same whether it comes from a cheek swab, a blood sample, semen sample, hair swab. There are certain very, very rare medical conditions where somebody might have a slightly different DNA profile. So this is something called a chimera. And also if someone had a bone marrow transplant, say following leukemia, the blood DNA profile would be that of the donor rather than that of the person. So it means the cheek swab will be different to the blood sample. But it's very, very rare, and it's normally something that we know about. But generally, in a rape case, you only have to give a mouth swab. You should not be required to give a semen sample. Now, how likely is it that somebody else would have the same DNA profile as me? So this depends on a number of factors, things like the size of the population that you live in and how many relatives you have. But if you talking about unrelated individuals, so the probability that somebody else unrelated to you has the same DNA profile as you is said to be around 1 in a billion. However, if you have lots of brothers and sisters, then the likelihood of somebody else having the same DNA profile as you drops about 1 in 10,000. If you've got an identical twin, and then yeah, somebody else will have the same DNA profile. But unrelated is 1 in a billion. And both siblings about 1 in 10,000. An identical twin is 1. You're gonna have the same profile. What kind of errors can occur with DNA profiling? Typically, not that many these days. I mean, when DNA profiling came forward, it had a bit of a baptism of fire. So constantly being challenged. So as a result, it's now very, very rigorous. So the kind of errors that can occur tend to be things like contamination, somebody somewhere cut a corner somewhere or something, or not quite pipetted it correctly. So I think probably the biggest error when it comes to DNA profiling is interpreting the significance of a DNA profile. So the DNA profile matches, brilliant. So what does that mean? So if you've got a DNA profile from a semen in a rape case matching that of Joe Bloggs, does that mean that Joe Bloggs is the person who raped that woman? And I said, well, what's the relationship between the two? Well, Joe Blogg is married the woman, then a consensual sexual relationship. The significance of that DNA is nothing. If, however, Joe Blogg and the victim were strangers, then it becomes very significant. So even though the DNA profiles are the same, the strength of it is very, very subjective, depending on the case circumstances surrounding it. So, in my experience, that's where the error in DNA profiling lies in, is how it's interpreted and how it's presented within the context of the case, rather than with the DNA profiling process itself. So the DNA profiling process itself is quite robust, and providing all the procedures are followed correctly, very little errors really occur. It's more the interpretational side of things. So what is forensic genealogy? So this is a fairly new term that's come out in relation to the Golden State Killer or Garden State Killer. So it's a place in America where somebody was identified using genealogy websites. And what this is, it's the same process as familial searching, where we can use the DNA database to identify your relative. But what's different about forensic genealogy is rather than using the National DNA Database, which is a very highly regulated and controlled database overseen by the government, these databases in forensic genealogy are commercial databases. So if you ever go onto these sort of ancestry websites where you want to find out who your ancestors were, in other words, carrying out genealogy, and what they do, they get your DNA sample in, they can tell you the ethnic makeup of your background. So you've got your DNA profile with the commercial companies. So what's been happening, the police have got a DNA profile from the crime, uploaded to the state national DNA database and found no matches, and then they've gone to the commercial DNA databases and looked on there. And that's how they found the Golden State Killer, is looking at the commercial databases that's available in America. And they were able to identify them after several years. So, but the initial science had been around in 2003. However, what's different about this is the database that's being used. So rather than using a national state DNA database, they use a commercial database. And the issue with that is it's far, far less rigorous, far less regulated. And also, what's happening, people uploading their DNA samples or uploading their profiles, not knowing that their profile is going to be used for a forensic investigation or for legal enforcement purposes. So that's where some of the criticism and concerns are coming around from forensic genealogy. But the science itself has been around for ages. It's just what database is being used. So I think those are the most common questions. So if you do have any more questions, then please do ask. I do like to get these questions, and the more off-the-wall questions you can think of, the better, really, I think, for me. So if you want to do that, you can just visit this website, and what we have is our forensic advice and consultancy service at Staffordshire University, and there's an inquiry page. So go onto that, you pop your question in there, and we'll get back to you with an answer. Okay? So I hope you've enjoyed that. And like I said, if you do have any more questions, just let us know. And thank you very much for listening, and enjoy National DNA Day. Okay, bye.